Today I'm going to be doing a training on TCR and BCR sequencing. A couple months ago, I did another training on TCR and BCR sequencing, but that was focused on 10x Genomics single cell platform. And so today I'm pretty much going to cover everything else that we offer, bulk TCR, bulk BCR, and the Takara single cell kit. So as a reminder, I'll be recording the training, and all of these trainings can be accessed in LIMS. If you go to the sales tab in LIMS and then click on technical FAQ, and then you have to go to the training tab here. That's a bit hard to find. But then you can see a list of all of the training videos that we've done. For the agenda, I'm just going to start by refreshing everybody on a few basics of immunology, specifically go over the structure and function of T and B cell receptors, and kind of go over the antigen receptor diversity, explain kind of why these are so interesting to scientists. Next, I'll go over the different types of methods for sequencing TCR and BCR. After that, I'll go into specifically what we offer here at MedGenome, the kits we use, the unique offerings, the different types of analysis, and then I'll also go over the single cell TCR at that point. And then lastly, I'll go over a couple of our competitors and the different types of TCR and options that they offer. And by competitors, I really just mean other providers of TCR sequencing, BCR sequencing, and that a lot of our clients are currently using. And I'll go over the pros and cons of each one so that you're prepared to have discussions with clients about this as it comes up. But to start, B and T cell lymphocytes are the major players in the adaptive immune system. Their receptors are responsible for pathogen recognition, and they play a crucial role in the immune response. As you can see here, the B and T cell receptors are heteromers. The B cell receptor consists of two parts, the heavy chain and the light chain. And the T cell receptor consists of two parts, the alpha chain and the beta chain. There are also T cells that have gamma delta chains in the T cell receptor. And I'll talk about that in a while. But for most of this training, you can simply think about alpha beta because those are the most abundant T cells and also the most relevant to most of our clients. So the thing that makes these TCRs and BCRs so interesting is that they're so highly diverse. And this allows them to recognize different types of pathogens or antigens. And this diversity comes from a few different mechanisms that I'll go over. So the first thing to show you here on this cartoon is this is a T cell with the T cell receptor. Here's the alpha chain, the beta chain. And the first thing you probably notice is that the beta chain is made up of an additional segment compared to the alpha chain. There's the variable segment, the diversity segment, and the joining segment, so VDJ. There's also a constant region, which isn't super interesting, but it is there. And on the alpha chain, you have just the variable and the joining, so V and J. So this comes from, so the first step in creating diversity in the TCR starts at the germline DNA. So for example, the germline beta DNA has multiple V segments that are non-continuous. It has multiple D segments and multiple J segments. And so these get recombined so that ultimately you end up with the T cell receptor that only has one V, one D, and one J on the beta chain, and one V and one J on the alpha chain. So just in the combination of which V segment gets combined with which D segment, with which J segment, that's the first step of creating diversity in the T cell receptor. The second step in creating diversity, it's called junctional diversity. And so when these segments are combined, when you combine this V segment with the D segment or with the J segment, at these junction sites, nucleotides are randomly inserted or deleted. And so that small change there adds for a little bit more diversity in the TCR or BCR. And then third, just the alpha beta pairing alone, or in B cells, the heavy and light chain pairing, provides some diversity. Because if you did happen to have the exact same alpha chain in two different T cells, you might have different beta chains. And so just that pairing alone makes it diverse. The fourth thing I have on this list is only relevant to BCR samples, but there's also somatic hypermutation. And so what that is once the BCR is exposed to the antigen, then you get all these extra mutations in the variable region that can lead to a higher affinity for this receptor antigen interaction. And so all of these, again, make this segment of the TCR or BCR interesting to researchers. Looking at the structure of the TCR and BCR, I found this cartoon online, which helps a lot. It shows these little blocks of that compose the BCR and the TCR. And you can see they're made up of these immunoglobulin domains. And these IG domains are made up of a couple of main parts. First are these anti-parallel beta sheets. These are kind of represented in this constant region of the TCR. They provide structure for the receptor. So because they're structural proteins, they're not very variable. They're highly conserved. And so they're not that interesting to most researchers. What is interesting are these loops at the end. These are represented in this cartoon by the VJ or VDJ segments. And these represent the CDR1, CDR2, or CDR3 regions, complementarity determining regions. CDR1 and CDR2 are simply found in the variable segment of the TCR or BCR chain. CDR3, that's the found at the segment where the VD and J segments are joined. And since I just explained all of these mechanisms that cause diversity at this region, that's the most diverse region, and a lot of times it's the most important to our clients for studying. Next, I'm just going to talk about the various ways that can be used to sequence these regions. On this diagram on the left, I have the smarter TCR and BCR kits. These are the Takara kits that we use in our lab. I'm going to discuss those in depth on the next slide. So for now, you could ignore this. It's here to just show the similarities between the multiplex PCR methods and to sort of contrast which segments end up in the final libraries. But these multiplex PCRs, we don't do these at MedGenome, but they are done at companies like Adaptive or iRepertoire. And it's a different strategy altogether to get these sequenced. The multiplex PCRs, they're done with hundreds of primers that bind to these variable regions, the joining region here. And you could use either DNA as input or RNA. For Adaptive, it would be DNA, and for iRepertoire, they have both. You could either use DNA or you reverse transcribe RNA and then use that cDNA as your input. But you can see from this, the positive of using DNA is that it's a lot more stable than RNA. And with this multiplex PCR, because there's only one copy of DNA in each cell, it makes it better at quantifying absolute values of certain clonotypes as they appear in your sample. Using RNA for both of these, they're not as easily quantifiable because each cell will have multiple transcripts of these TCR or BCR chain mRNA. And so you can measure relative abundance of these clonotypes, but you won't be able to tell absolute number of cells expressing these TCR or BCR. Looking at the TCR kit that we use in-house, actually a lot of the TCR kits kind of work on a similar principle. And whether it's the TCR, BCR kits or RNA-seq kits, but they use 5' race, which stands for rapid amplification of cDNA ends. And so what happens here, specifically in the TCR and BCR kits, is you start with a primer for reverse transcription, and this is an oligo-DT primer with a tag at the end that has a known primer sequence. And so the reverse transcriptase will, once this primer anneals to the poly-A tail, the reverse transcriptase will transcribe the cDNA until it reaches the end of the molecule. And this specific reverse transcriptase in the TCR kits, it then adds some non-template nucleotides to the end. And these exact non-template nucleotides happen to be complementary to a template switch oligo that's also in the reaction, which results in this template switch oligo annealing and then allowing the reverse transcriptase to finish the sequence, adding this additional non-template sequence to the cDNA. So you end up with a first-strand cDNA that's synthesized that has a non-template tag on the 5' end and a non-template tag on the 3' end. And since we know these, then we can use this to do PCR to amplify the cDNA. In a typical mRNA-seq kit or an RNA-seq kit, you would amplify all of the RNA. But in the TCR and BCR kits, we're really only interested in the alpha chain and beta chain for TCR or the heavy and light chains for BCR. And so PCR1 is where those chains get amplified with the first round of PCR. And then there's a second PCR that adds the Illumina indexes so that you can sequence them. A couple things worth noting here, just that some of the kits do have UMIs, unique molecular indexes, so you can help control for PCR bias, because there are a lot of PCR cycles that ultimately happen between the input and the final library. There's a TCR V1, the older TCR kit that it's been discontinued by Takara, but we still have a lot of it in stock in our lab. And this kit does not have UMIs. So that's just something to be aware of if you're discussing this with clients. Do they need UMIs? If so, make sure you steer them toward the V2 kit. And then just some things to consider when someone is trying to choose between using, say, the TCR kits that we offer, the 5' race method, or using one of the multiplexing methods. First, it's just, should they use DNA versus RNA as a template? So there are pros and cons to both. Using DNA, of course, like I said, can make it more easy to quantify. DNA is incredibly stable. This DNA allows for different types of input sources, so you can use FFPE and get pretty decent results. The downside of it is just that because there's only one copy of DNA per cell, if you don't capture something in your library, perhaps a small subpopulation of cells, then it's completely going to get missed. Whereas with mRNA, because there are so many copies of each transcript, you sort of have multiple chances to capture these rare clonotypes in your data. The next thing to look at is, is a client interested in just the CDR3 region, or do they need the full length VDJ section? So the Takara kit offers full length VDJ. And even beyond that, the Takara kit offers single molecule sequencing. So using this PCR, you amplify just that VDJ region, and you add these indexes to it, but you never fragment it, and then you're not reforming the molecule using bioinformatics. You're sequencing the entire molecule. So a general kind of rule is if the ultimate intention will be to clone either the TCR or BCR, then if they're going to be doing like antibody or T cell therapy applications, then full length sequencing will just make everything easier in the end. But a lot of times clients are really only interested in the CDR3 region, in which case the multiplexing method is just fine. And then lastly, something to consider is bulk versus single cell. And the main advantage here is just that with bulk experiments, you can analyze more cells. You can go on with thousands of cells, millions of cells, and find all of the alpha and beta chains or heavy and light chains. But because the cells are lysed and you use RNA as input, everything's mixed together, and there's no way to find the actual pairing. So again, the pairing of the alpha and beta chains adds diversity. There's no way to know the exact pairing in a certain cell. And so there are advantages to both. And a lot of times if a client isn't sure what they want, it's always good to start with a bulk experiment because it's relatively inexpensive. And then if they find the clonotypes of interest, then they could always follow up with a single cell experiment to find the pairing information. To cover the workflow of the kits that we use in-house, this is just a general workflow for the bulk TCR kit, and I'll do the BCR one after this. They're very similar in most ways. A couple things to note though is just for the input for the TCR kit, you can either use cells as input or you can use RNA as input. And a lot of times when clients are sequencing T cells, they don't get a lot of cells. If they're doing PBMCs as input, usually it's not a problem to get hundreds of thousands or millions of cells. But if they sorted their T cells, a lot of times it's pretty limited. And so with Takara's kit, you do have the option of using the cells as direct input by having them submit their cells up to 10,000 in about 10 microliters of Takara lysis buffer. The downside to that is there's no QC step until pretty much libraries are made or after the PCR step. So there's no way of knowing if this will work. Perhaps their cells weren't viable when they added them to the lysis buffer, which has happened on previous projects. If a client has more than 50,000 cells, then we always recommend just doing an RNA extraction and using the RNA as input. And if they have somewhere in between 10,000 and 50,000, it gets a little tricky. I always recommend just aliquoting 10,000 cells and sequencing that. But if they have like 40,000 cells, we could always try the extraction. In my experience, it's just hit or miss whether we'll be able to detect the RNA after. And so we might not be able to confidently make a library. But using the RNA as input, I put a note here, high quality RNA is the best, because again, going back to the fact that this is a single molecule we're sequencing, it makes sense that it helps to have that molecule intact. If the RNA is degraded, then there's a chance that the VDJ region will be degraded, and so we might get truncated clonotype sequences. or the poly-A tail could be degraded, and we'll just have very inefficient reverse transcription. The other thing is to use as much RNA as possible. And I have a figure showing this a little better in a few slides, but when you're, again, a lot of projects you're limited on how many cells or how much RNA the client has. But if a client does have, micrograms of RNA, it's always good to use as much as possible because you get the most diverse libraries that way. But that being said, it does work with low quality RNA. We've used it for FFPE, and it also works with low inputs. The kit recommends 10 nanograms input, but we've certainly used it with picograms of RNA and still got sequences. So it's pretty robust. As far as libraries go, by standard, we generally provide both alpha and beta chains for each sample in a single library. But these primers are in separate tubes. So if a client isn't interested in alpha chains, for example, we can make the libraries using only the beta chains. Or if they want to make sure they get a lot of alpha and beta chains represented in their data, we can always, make two libraries, one for alpha chains, one for beta chains. So there are options here to give clients the best data. And I also included some QC for FFPE. Again, FFPE, you're going to get lower quality RNA as input. And so instead of seeing these nice clean peaks for the library profiles, you get more of a smear. And this is actually one of the better FFPE QCs also. A lot of times it could just be a large smear. And so, yeah, and I'll cover the FFPE in a few slides. When it comes to sequencing, this has to be done on MiSeq because you're doing these, you're sequencing these single molecules. You need the longest reads possible. So Paradigm 300 is what we use. As far as sequencing depth, it's a little variable, and it depends a lot on the input. 100,000 reads will certainly get you some good data. Based on the input used, if you use only a few nanograms of input, 100,000 reads will probably be more than enough. But if you used micrograms of input, hundreds of nanograms, then you may want to sequence up to more of 1 million reads for samples, because again, it'll get you more data, more clonotypes detected in complex samples. And then one other thing to mention for sequencing is that ideally we want to pull similar samples together when sequencing. So keep the TCR samples together, keep non-TCR samples together. Because these libraries are so large and they have a lot of PCR artifacts that won't exactly cluster, it's hard to say how well these will cluster and how much data, and it's hard to predict how much data they'll actually generate. Whereas other types of libraries, your typical RNA-seq, amplicon libraries, it's pretty straightforward. They generally cluster extremely well. So if you try to mix these together with smaller libraries, you'll get data, but you may not get the targeted amount of data. So it can be done, but it should be done only as needed. And then lastly, I'm going to cover these individually, but we do have a lot of TCR and BCR analysis options available for clients. Standard options are actually really good and are kind of industry standards. We also have some advanced options available, which are a bit more niche depending on what the clients are looking for, but we have a lot of these available, and our analysis options are interactive on our Mingo platform. For BCR, it is a lot of the same, but I will cover a couple of things that make it different from TCR. And the first thing is just that we can only use RNA as input for BCR. We can't use cells. The other thing that's different is that the amount of libraries you end up with per sample is variable and depends on the project and what the client's looking for. So each Takara BCR kit, it comes with 4 sets of primers to amplify the heavy chain IgG or IgM, and then light chain IgK or the kappa and lambda. And so there are options here. The client, if they want to see data for all 4 of these, we can make 4 individual libraries, amplifying each of these individually. We can combine the 2 heavy chain and the 2 light chains together and make 2 libraries per sample. Or if they're only interested in some of them, we just make what they're interested in. We have tried combining all 4 together in a single reaction for one client, and I think it was fine. I think it worked, but it's not recommended. So this is important to have this conversation with your client when you are first setting up the project, because of course each of these libraries that you make will be sequenced individually, and so you should, the amount you charge changes. So it's not really charging per sample, but you're charging per library. And then a lot of the sequencing and analysis are very similar. Next, I'm going to talk about some of the things that MedGenome offers that really make us unique in the TCR market. And the first thing to discuss would just be gamma delta T cells. So I mentioned them very briefly at the beginning of the training, and I said that the gamma delta T cells, they do exist, and that they're at a much smaller abundance than alpha beta T cells. And so the TCR expressed on these cells, they have gamma chains and delta chains instead of the alpha chains and beta chains. And these cells, their role isn't, their role isn't too well known, but just some of the things that have been documented is that the gamma delta T cells play more of a response, sort of linking the innate and adaptive immune system. They can act as early responders to pathogens such as viruses or bacteria. They can be found in tongue, uterus, placenta. They promote tissue repair and wound healing in the epidermis. So they're they kind of serve very unique functions. And a while back, Ahmed's group has designed primers that we can use with the standard Takara kit to amplify the gamma delta chains. So this is something that we developed in-house. There's validation data for it. So it's not something that most other companies are able to do. As a note here, we've only ever tested this with the TCR V1 kit. Again, we do have this in stock until it's depleted, but Takara has discontinued this. So at some point we'll have to test it with the TCR V2 kit. I expect it'll work, but we still need to test it. And if not, it should be somewhat easy to design these new primers that'll work with the V2 kit. And then another thing to mention is that we currently only have 2 sets of I5 indexes for these gamma delta PCR. And so for each chain, there's I5, 502 and 503. And then there are 12 possible I7s. So for each one, there's 24 combinations of indexes you can use. We've had several gamma delta TCR projects in the past. Most of them are pretty small, but if we ever do, need to scale up for some reason, we have these primer sequences. It'd be easy to order new ones and just swap in new 8 base indexes. Another thing that we offer that makes us somewhat unique in the TCR, field is FFPE TCR using RNA as input. So here, a lot of the optimization, the kit being used is still the Takara kit, but a lot of the optimization was done in the amount of PCR cycles that we use, the initial primer for reverse transcription, And then really a lot of the optimization was done on the analysis end in order to take this low quality data and still be able to get meaningful sequences from it. A couple of things here to mention is that we do offer this, and it is certainly, it has produced useful data for some projects, but it is very hit or miss in that it's the quality of the data that you get is really dependent on the quality of the input. Every FFPE slide is different. Every section of tissue is different. And if there's not a good representation of T cells on that tissue, on that slide, then it's not going to show up in the data. Another thing to keep in mind is just the quality of the RNA that you get from FFPE. The quality is going to be pretty bad regardless from FFPE, but even within the low quality RNA that you would expect, there's still a range of DV200s that you might get. And I included this figure at the bottom that comes from our white paper, but you can see that as the DV200 decreases, you see a significant decrease in the amount of total unique CDR3s detected as well. So, you know, the worse quality the RNA, the fewer clonotypes you're gonna detect. And again, this all comes back to what I said about it being a single molecule sequence. Degraded RNA is not going to be efficiently reverse transcribed, and a lot of your sequences may be truncated. One third thing that helps us to stand out in the TCR field is involves the species we can use our TCR kits with. And so Takara, the actual kits that we buy from Takara are only compatible with human and mouse species. And looking at Adaptive and iRepertoire, they're also only offering human and mouse. But our bioinformatics team, specifically Kushal, has worked with a client in the past who was studying cynomonkeys. And so Kushal designed these primers that we've used to amplify the TCR in cynomonkeys. I think we've used them with other types of monkeys as well, but the point of this is that this is something certainly that can be useful to a lot of labs who don't use human or mouse as their model organisms. Even if they're using something else besides monkey, it's possible that Kushal or his team can design primers to amplify the TCR chains in these samples. It really depends on how well annotated the genome or transcriptome is for the species they're studying. But if you do find a client who's working with a unique model organism for their projects, it'd be a good idea to bring in our informatics team and see what the options are. So next, I'm going to discuss some of the analysis options that we have that we can provide for TCR and for BCR. So the first thing that I'll show is just these spectrotype plots for CDR3 length. So these are plotting a couple of things. It's plotting the abundance of the various lengths of CDR3 regions in each sample in the sequencing data. And then it's also showing the most abundant clonotypes where they fall and in these different lengths. And so what you would expect, these are taken from some client projects, but on the left, you see just a healthy individual PBMC sample. And so what you expect to see is sort of a bell-shaped curve here, representing the various sizes of CDR3 lengths. And the one on the right, this is a sample taken from tumor RNA. And you can see you sort of lose this bell curve because some of the lengths are very highly expanded. Some of the clonotypes are highly expanded. Um, and so this is useful because it gives a really quick overview of what type of data you can find in your samples. Just by seeing this clonal expansion and the CDR3 length, you know that there's something interesting going on in your samples. Next figures are these VJ gene usage Circos plots. What these are showing are just the the V and J genes that appear together in the same sequence data. So if you follow this circle around, I know it's hard to see on this figure, it lists all of the alpha V genes and all the alpha J genes, and then all the beta V and all the beta J genes. And so for each sequence that we in the data, it's gonna, like I said, there's gonna be one V segment and one J segment represented. And so each string just shows that V and J pairing that appears in your data. And the more abundant that pairing is, then the thicker these strings are going to appear, the thicker these bands are going to appear. And so this first one, this is just a healthy individual PBMC. And so you can see, nothing really stands out here. Of course, there are some bands that are thicker than others, and this could just be, the individuals themselves. It could be, whatever's going on in their system. It doesn't always mean that these are extremely interesting, but if you do see, but then if you look at a tumor RNA sample, you start to see, some segments, some V and J segments are a lot more abundant. And so you can, you kind of zero in on trends there that could be caused by the tumor. Yeah, I'll move on. Sorry. And then, so the next figure I wanted to show is this Shannon diversity index plot. And so these plots will show you relative diversity of the clonotypes found in your samples. So I have a couple of examples here. And this example on the left is more of a technical example. This project was done with the same PBMC sample, and it was different inputs were used, 10 nanograms, 100 nanograms, or 1,000 nanograms of RNA. And it was done in duplicate. And then a separate sample, a positive control, was also used with 10 nanograms. But the takeaway here is that using the exact same sample, the more input you use, the more diverse the sample is. And so again, it comes down to if a client has micrograms of RNA available, it's always best to use the highest input for making these libraries versus using the lowest input, because you can see there's quite a bit of a difference between a 10 nanogram input and 1,000 nanogram input from the exact same sample. And on the right, we just have sort of this is more of a technical on the left, a technical example of the Shannon diversity index plot. On the right, it's more of an experimental use for it. And you can see the positive control here, which is just a PBMC sample that comes with the kit, compared to some stimulated samples. And of course, the stimulated samples are all going to be less diverse, or you would expect them to be less diverse than the positive control. And you can see the highly stimulated are even less diverse than low. And the last figure I wanted to show is just this pairwise scatter plot. And what this is going to show is it's going to show the shared clonotypes between your different samples in a project. So what this figure shows, and it's kind of hard to see here, is that the clonotypes from one sample, for example, this PBMC 10 nanogram one, are here in blue, and the clonotypes from a second will be in yellow. These are the unique clonotypes. This is the positive control. And any shared clonotypes between the samples will appear in red across the middle. And so this is that same project I showed on the previous slide where these PBMCs were 6 libraries were made in replicates with different input amounts. And then it's compared to a positive control, which is also PBMC sample, but from a completely different source. And so you can see, as you'd expect, when you compare these PBMC samples to other replicates, most of the clonotypes are shared. But if you compare the PBMC from one sample to PBMCs from the positive control, there are very few shared clonotypes, and most of them are unique. And this is actually really useful for a lot of clients are looking for, maybe common clonotypes amongst different samples, different tissues, different patients. So this is a really great way to capture that. And then also the statistics are here on these side boxes. So before I wrap up, I think they're out of order, but that's fine. So one other thing I was going to discuss was the single cell TCR kit from Takara. We do, last time I talked about all of our 10x Genomics single cell offerings. Takara also makes a single cell kit, which is a plate-based sequencing kit. There are a few restrictions on this. It is human only, but it does provide alpha beta pairing. It is not as scalable as the 10x Genomics single cell TCR, but it's perfect for clients who don't have a lot of cells. And maybe they just need that alpha beta pairing from a small population of cells. Then they can sort their cells into a plate and send those to us. For this TCR plate-seq, there is a preferred plating method for the client. They should leave some wells empty for us to add negative and positive controls. But for the most part, it's pretty straightforward. And we have protocols to share with them for that. Oh yeah, one other thing about this kit is that it's actually fairly expensive. For a plate of 96 samples, I think it would come out to a few $1,000s for it to be sequenced, which is kind of a difficult sell when you compare that to a 10x project, which you can get hundreds or thousands of cells sequenced for maybe just a little bit more money. And so one thing we hope to do in the future is to see if we can get this kit to work at a lower volume than what the kit recommends. So for example, the single cell RNA-seq kit that Takara makes, you can buy a 96 reaction kit and you can make the libraries using the full volume that the kit specifies. But what we've done is we've used our Mosquito, which is a low volume dispensing machine. And we're able to cut the volume used to quarter volume, so 1/4 of the volume. And that allows us to turn a 96 reaction kit into essentially a 384 reaction kit and lower the cost to make it more affordable to our clients. So hopefully what we'll do in the next few weeks or months is try the same thing with this single cell TCR kit and see if we can get the same quality data from these single cells if we cut the volume in half or quarter or something. Because even, if we're going from $3,000 a plate or whatever to $1,000 a plate, that can make a huge difference and make it, a lot more sellable, a lot better option for clients. And the last slide I'll go over is just a summary of some of these TCR providers that exist. These are the two that come up a lot in conversations with clients. And so I wanted to summarize some of the information about these, their offerings. On the left is adaptive, and they have the immunoseq platform, which is DNA-based multiplexing. As I mentioned earlier, it only captures the CDR3 region. It's not full length. It does work well with FFPE samples. They do full service TCR sequencing, so you send your samples to them. It's not necessarily like a kit you can buy and do it yourself, unlike Takara. The price is about 500 to 1,000 per sample, but this also gets you access to their immunoseq analyzer, which is their interface for analyzing your data. And you can also get access to the Immune Access TCR BCR database. So, we come across a lot of people who use this. It, again, has a lot of pros and cons. If we were trying to convince someone to use the Takara method that we use over this, I would really emphasize the full length VDJ region that we'd be providing. That's not always interesting to clients. Another thing, but we can capture it regardless. Another thing we have is more flexibility with pricing. 500 to 1,000 per sample can be a bit steep. And the kits that we use there, we do have flexibility for some clients to lower the price if they're analyzing a lot of samples in the same project. And they have their immunoseq analyzer. We have our Mango platform with interactive analysis figures. So there's some level of similarity there as well. So I think for some clients, you may be able to convince them to switch. Others, maybe it's just, it might be a hard sell. iRepertoire is another one that comes up a bit, and they are certainly, they have a wider variety of offerings compared to adaptive. They also use a multiplexing based method, but they use both DNA or RNA as input. And based on how their primers are designed, they capture the CDR1, CDR2, and CDR3 regions. It does work well with FFPE. Their species are human and mouse. They do have plate-based single cell options called iPair. And for their bulk TCR, I know the lowest you can get is $300 per reaction, and that gets you a single chain sequenced. So maybe an alpha chain or probably a beta chain. And it just gets higher from there. They have bulk BCR, they have gamma delta TCR also. So that one's actually a lot more comparable to what we do as far as deliverables go. Again, where we sort of stand out is just the platform we use offers full length VDJ sequences, the ability to possibly design primers for other species that could be helpful here. The pricing that iRepertoire offers is somewhat competitive, but the $300 buys you only a single chain of TCR data and does not include analysis. So there's still some flexibility there to, sort of compete on pricing and the analysis options as well. So, but that's everything I have. Are there any questions from anybody?
Yeah, I guess my first question is about the V2 kit. Oh, so you mentioned that, if I understood correctly, you mentioned that Takara is discontinuing the V1?
Mm-hmm. Yes.
When are we fully switching to V2, and will there be a price difference?
So I'm pretty sure Takara has stopped selling V1 because I went to their website, I think yesterday, and I think it redirected me to the V2 kit. But we still have probably dozens. I'd have to ask the lab to do an inventory, but I think we have dozens or hundreds of reactions still in the lab. So we be using those until it's depleted. Anytime a client doesn't specify they need UMIs, then that would be the option we would use. And then as far as switching completely to V2 or the price difference, I think the kit is, so back when, a few months ago when I ordered the kits, the TCR V2 kit was a little bit more expensive. And I want to say when we did a calculation, it was probably like an extra, $10, $20 more per sample, but you'd have to double check that in LIMS. There is a slight price difference. It's, since then, Gil has talked to the Takara people and renegotiated our discount list. So it's very possible also that he got it at a similar price to the V1 kit. So double check LIMS. I know that's been updated by Vinay. So it should be right.
Hi, Olga. I just double checked and both kits are under the library prep option and it's $55 more expensive.
Perfect. I remember it being more expensive in the V1. Okay, perfect.
Thank you.
But we'll see how that, like what ends up happening is every time they order it and we get new discounts, the price does change real time. So in the future, it might be less.
Thank you.
Hi, Derek. This is Chaitanya. Thanks. It was very detailed technical presentation, like explaining the advantages over DNA as an input and RNA as an input, or CD or full length or single cell versus bulk TCR. I have a question on the inputs wise, irrespective of going lower input, but it has to be quality RNA. But how reliable results are when you have very low minimum inputs or degraded RNA from the FFP? I mean to say, is there any challenges in order to get low amendment TCRs or rare clonotypes? What is your recommendation?
Well, so using low input, high quality RNA, we've certainly gone, the kit is meant to go down to, I think, 10 nanograms as the lowest. We've gone down to like 1 nanogram, certainly using the V1 kit. And using the V2 kit, one of Angelica's clients, we were able to do, a few hundred picograms, and we got, thousands of clonotypes for each sample. So the kit itself is pretty good when the quality of the RNA is good. Once the quality gets poor, and I know Gavin's done a lot of FFPE TCR libraries, it's so hit or miss. He's, ideally with FFPE, we'll also have a lot of RNA, but we've done some with low quality, low input, and the results were really terrible. And that actually is true just for low quality RNA that's not FFPE. You still get a library, you still generate data, but the data itself is just, it's not good. So when it comes to a non-FFPE sample, I would try to stick close to what the kit recommends, the 10 nanograms input, high quality. If you don't have that, I would always have that discussion with the client first before proceeding, because if it's really low quality, low quantity, especially, we'll generate libraries, we'll generate data, but it'll just be garbage.
Sorry,
Greg,
can I
I
ask
have
one
a
more?
question.
Oh, go ahead.
The UMI kit I know comes with the human or mouse, I forget.
Human only.
Human.
I think for TCR, it's certainly human. For BCR, I think it's also only human, but I would have to double check that.
But is there an option to have a non-UMI version?
Well, for TCR, it's only that V1 kit that's been discontinued. So they're pretty much phasing that out.
I see.
Yeah, and then for BCR.
At some point I remember reading a review in Nature Biotech, and I can share the paper with the team, and this might be useful for questions that might come up. It's shown that the UMI method is actually less sensitive compared to the non-UMI method for capturing the clonal diversity. So, in case the question comes up, it's useful for distinguishing between clonotypes, obviously because of the UMIs, but it's apparently less sensitive. So just so that everyone's aware. And I can share that paper. I'm trying to find that.
Yeah, that would be great, actually, because I think it does come up quite a bit in conversations.
Yeah.
And it is hard to, especially if we have these leftover TCR B1 kits, it's hard to
Right.
convince somebody other than price to use that.
Derek, I just want to thank you for a very informative session, and especially going through bulk versus single cell versus towards the end, going through even the adaptive versus eye repertoire differences. I think that's really helpful. So, you know, not just the slides, but also the recording will be immensely helpful. So thank you again for doing this.
Well, thanks for saying that. And thanks everyone for joining. Are there any last questions? Otherwise, I'll wrap up.
Thanks, Doug.
Okay, awesome. Everybody have a happy Friday.
Thank you, Derek. You
Thanks,
too.
Derek.
Thank you, Derek.